So in this little lesson, I'd like to look at neurophysiology. In previous videos, we have seen what happens between a neuron and a muscle. We talked about a synapse called the neuromuscular junction. And in this case, we see a synapse between two neurons, a neuron-to-neuron synapse. So it's pretty simple that we have a presynaptic cell and a postsynaptic cell, and the presynaptic cell and postsynaptic cell both are neurons. The neurophysiology is a little bit more simple than it actually is in a muscle cell. Now, when we look at this, we are going to deal with the same sorts of ions, potassium cations and sodium cations and calcium 2 cations. And those three major players all come into play very similar to what we've seen before. Now, as we look at this, if we begin our cells with the presynaptic cell, for more simplicity, we discussed in the neuromuscular junction that there were vesicles full of neurotransmitters called acetylcholine. Well, now we're not so concerned. I'm going to use a different color, and we're just going to say these are neurotransmitters. It could be oxytocin, dopamine, octopamine, whatever different kinds of neurotransmitters we may use, different classes. So in here, we're not so picky as to what we're looking at. Now, one of the things I always like to kind of start off with is we want to get this neuron here into a resting state. Now, the presynaptic neuron, it's resting, it's ready to go. We can already think about it as resting. But how does a neuron get to resting? Well, just like a muscle cell gets to resting, we use a protein drawn in blue here on the postsynaptic cell called the sodium-potassium ATPase pump. The sodium-potassium pump will bring in 2 potassium cations and stick out 3 sodium cations, and this will bring it to resting. Now please note that the inside of the cell is slightly negative and the outside will be a little bit more positive in respect because of the distribution of these two ions and their charges. So a resting cell can be thought of as being polarized. Now, do we see why it's polarized? Because the charges are opposite on the inside and the outside. So as this cell is now polarized, think of it as cocked. Our action potential, which I did not mention in the previous video, is an all or none event. So this will either go or it won't. And how we're going to get this to happen can be quite an interesting thing. So now we've gotten to resting, we're going to remove our sodium-potassium ATPase pump and remind ourselves at this point, while it is resting, that this cell has a high concentration of potassium inside the cell and a high concentration of sodium outside the cell. And as this occurs, we already remember that an action potential propagates down a neuron. Drawn here is what looks like a lightning bolt. This opens up the voltage-gated calcium channels, and calcium, Ca2+, miswrote it there, Ca2+, will come in. Now this results in the vesicles fusing with the plasma membrane. Because they are vesicles and come off the Golgi apparatus, they are made of the same material as the plasma membrane, and they will exit via a process called exocytosis. Now, when neurotransmitters come, they cross the synaptic cleft. When they come out, they cross the synaptic cleft. Now, on the postsynaptic cell, on the what we call the postsynaptic membrane, we have receptors. And again, I'm going to draw it just like I did in the past, little Y shape, little guys, little Y shapes. And you will see whatever neurotransmitter I'm drawing, I drew them in orange and I changed colors. But if they bind to the receptor sites, these will open up our sodium gates. Sodium gates open. So I'm going to color in a sodium gate here, and sodium Na plus rushes in. Now note this cell was negative. Now here's the interesting thing. When a neuron is at resting, it is at negative 70 millivolt. This cell will go to negative 60. Now, as sodium rushes in, anything lower here, anything when we go to negative 60, when we get into the 60s, essentially we are going what we call threshold. So negative 60 would be the threshold level as we begin making the cell slightly positive. As we make it positive in result to the outside of the cell, we call it depolarized. This cell is depolarized. You see why it is depolarized? Because it is the same charge as the outside of the cell, as it is inside the cell, as it goes to negative 60 millivolts. Now, we will change the charge to another level as we get more and more positive. If we open enough sodium gates and we let in enough sodium, we will go to a height of plus 30 millivolts. Now notice negative 70 and we start going closer to 0, negative 60. If we make it to positive 30, we are firing an action potential like a snap. Pow. The cell then fires the action potential. And once this happens, we will use some sort of enzyme to clean off all of our little receptor sites. So I can erase neurotransmitters and we can just get rid of the receptors. Our sodium gates will close. And as this happens, we will then become, now that we're depolarized, we shut those gates off, stopping it from getting further positive, and then we're going to open a new gate. When the voltage changes, we open up the potassium gates, voltage-gated potassium channels. Potassium will leave, K+, leaves, again, high to low. As it leaves the cell starts getting negative. So this is how we repolarize a cell. That leaves, and then we will start going back down, negative 60, negative 70, and we plummet down. It's about negative 90, and then we'll pop back up to negative 60, 70. And how we get there is with our sodium-potassium pump. Sodium-potassium pump here. And my sodium-potassium pump, again, 2K+. And let me delete some of these little ions so we can see better. And 3Na+. And this sets it back to resting. So we can get back to resting this way, and the cell can be back where it is negative, resting. So that should help you guys understand what is going on and how we generate an action potential in a neuron. And we don't have all of the confounding T-tubules and triads and terminal cisternae like we had in the muscle cells. It is actually quite a bit simpler. And this concludes my little lesson on the physiology of a neuron. Thank you.